So we're going to review our scientific method and look at some examples with the vocabulary. So yesterday we looked at these notes, and we also did a click and drag looking at identification of different steps. Notice that we start with either making an observation or asking a question, which leads us to do some research. So sometimes the steps may be in a slightly different order, but basically we're doing some background research because of a question or observation that we've noticed that helps us form our hypothesis. We're then testing our hypothesis and then finally collecting data and reporting those results unless we need to redo the experiment. So notice there's sometimes a slight change, but that depends on the specifics of the experiment. So we start with the observations that we gather using our senses, and we look at research to help us form our question. This is going to lead to our educated guess or hypothesis, which is written in the form of if and then, and includes the independent and dependent variable. This is going to help us give us a very specific prediction of what we think the outcome will be and why. The variable has two parts. The IV, or the independent variable, is what I change. It's the one part of the experiment that's manipulated. The dependent variable is what changes as a result of that. And so it's what's measured. When we set up our experiment, we do this to test our hypothesis, and it has two parts. There's the experimental setup and the control setup. In our example we're going to look at today, we have two plants, one that's the control and one that's the experimental. And I want to know what happens if we add energy drink to a plant. So that energy drink is going to be added to the experimental setup. And I want to know, does it actually impact its growth? So in this example, the energy drink is the independent variable. It's the one thing we're changing. The growth of the plant is what we expect to change as a result of adding the energy drink. And everything else about the plant, temperature, light, water, the type of plant, container, soil, will be called constants. And they're going to be the same in both plants. So we can see that the energy drink is only going to be added to the experimental setup. That's our independent variable. And our constants are controlled variables. The temperature, light, water, the type of plant, container, and soil will be the same in both setups. The purpose of the control is just to be an comparison to the experimental setup. It does not have the independent variable or the energy drink like you see here. And it's just going to show us that we got our results because of the energy drink and not because of something else. All of the things that we just talked about, temperature, water, the container, the soil, the type of plant, those are called constants or controlled variables. They will be the same in both setups. Our independent variable, remember, will be the energy drink. The growth will be the dependent variable. So looking at our experiment, if we want to write a hypothesis, we can look at our independent variable and our dependent variable. Remember, we want to phrase it in the form of an if-then statement. And we could say, if I add a can of energy drink to my plant each day, then the plant will grow taller. Remember, we would want to have several trials in order to make sure that we did, in fact, get our results as a result of the energy drink and not just due to chance. And remember, we have those two groups, the control and the experimental group, with only one variable, the energy drink. We would collect data. In this case, the growth would be quantitative. It would be organized into charts or graphs. And the data, we could make our conclusion for our experiment. We could retest to verify our results. And then finally, form our conclusion and publish our results.